de nosotros, de nuestra especie humana. Y bueno, sin más preámbulos, también damos la invitación al doctor Michael Ceballos de la Universidad de Arkansas. Él ha estado trabajando con la Universidad del Atlántico, tiene buenas intenciones con la parte académica y estudiantil de esta institución en cuanto a movilizaciones internacionales, siempre en la búsqueda de compartir el conocimiento, por lo que ya es un amigo de la casa de la Universidad del Atlántico y le damos la más cordial de las bienvenidas. Nos permitimos leer, él es el doctor en microbiología y bioquímica de la Universidad de Montana, con énfasis en ecología de enfermedades infecciosas. Muy buenas tardes y bienvenido.
Michelle Ceballos, profesor en el Departamento de Ciencias Biológicas de la Universidad de Arkansas.
Muy buenas tardes a todos. Voy a hablar inglés porque mi español no es tan perfecto y con las palabras técnicas a veces equivoco mucho. So I'm going to speak in English, but I'll speak very slowly. And si tienes preguntas en español, pues puedo manejarlos. So thank you all for being here today. It's exciting to present again at one of the meetings for the Astrobiology Institute of Colombia. The director, Jorge Bueno Prieto, is a close colleague of mine and a very good friend. He's just left my lab not too long ago. He's doing some research in my lab on extremophile microorganisms, and he asked me to give a talk today, and I'm happy to do so. So my talk might be a little bit different today. I'm not going to spend a lot of time talking about a very specific organism. I will do a little bit of that, but what I would like to do is talk a little bit more general about something that is important to us as humans when we consider space exploration. And it's a consideration that has piqued the interest of a lot of the federal funding agencies in the United States, and that is the nexus of food, energy, and water systems here on Earth. And what I would like to do is talk about that nexus of food, energy, and water systems and the innovations at the nexus of food, energy, and water systems, what they mean to us here on Earth, and then how it merges with and basically integrates with the astrobiology community. And so I do have a slideshow presentation here. It's way too long for the time that I have. I always have a lot more to say than what the time allows. But what I would like to do is basically split the talk into 3 sessions. I would like to talk about first, what it means, what INFEWS is, innovations at the nexus of food, energy, and water systems. And then I would like to talk about how that applies to astrobiology community. And then third, I would like to talk about research opportunities for students and faculty through collaborations with the Astrobiology Institute of Colombia and my active grants, most of which are funded by the U.S. National Science Foundation. So if you don't mind, I would like to share my screen with you at this point. And during the course of the presentation, I will pause and take a few questions. And then of course, there'll be questions and answers at the end of the talk. So can everyone see this? Can I just get a thumbs up or a yes or?
Yes, Michael, we saw.
Excellent.
Okay,
thank you so
see,
much.
perfect. Thanks so much.
All right. So we have a new award. It's funded by the U.S. National Science Foundation. And the title of this award is Innovations at the Nexus of Food, Energy, and Water Systems. Should be an S there. Brazil, Argentina, Chile, Colombia, and the U.S. So it's our Bacchus Research Network. And what I'd like to do is talk a little bit about what INFEWS means. So INFEWS, Innovations at the Nexus of Food, Energy, and Water Systems, and how that topic integrates in with what we would like to do in astrobiology. And so that's what I would like to do today, and also talk about research opportunities and then have a question and answer session at the end. So let me see if I can get this to go. So this is what we would like to do today, and hopefully we can get through that here. And I believe, what is the time frame? How many minutes would you like to spend on this?
You have 40 minutes.
How many?
40.
40. Okay, great. Yeah, that's perfect. All
40
right.
minutes, and then if you prefer, we can make the questions. So could be, if anyone try to make the questions in Spanish, I'm trying to traduce you or speak with you in Spanish the question you asked.
Ya, las preguntas se pueden preguntar en español o usar el chat. Y si puedo leer lo que están preguntando, también puedo responder.
Great, Michael. Thanks so much.
Okay. All right. So here is our topic, innovations at the nexus of food, energy, and water systems. And, you know, in the astrobiology community, most of us that are focused in astrobiology, we're always thinking about, you know, is there life on another planet or a moon, say, you know, one of the moons of Jupiter or the moons of Saturn, or is there advanced extraterrestrial life? Is there microbial life on Mars or even the moon where liquid water is found? And these are definitely important questions. But we also have some significant problems that we have to deal with here on Earth. And one of the most significant problems is the growth of our global population. So it's estimated by the year 2050, we are going to exceed 9 billion people in the world, which means that we're going to have about 60% higher food requirement, 80% higher energy requirement, 50% more fresh water withdrawal from the developing nations, even by 2025, and approximately 20% more fresh water withdrawal by the developed nations by 2025. So by 2050, these numbers will go up even more. What this means is there's going to be an increased demand for food, an increased demand for energy, and an increased demand for water. Coupling with that, we have climate change. So there's noted variations in the amount of precipitation that we're getting in different parts of our world. And we also see fluctuations in temperature, including some warming in certain areas of the globe. And so if you take a look at this increased demand for food, energy, and water, and the changes in climate, that's going to put severe pressures on our resource availability. What that leads to, in part, is a shortage of food, energy, and water, but other impacts, such as an increase in the prevalence of infectious disease and pandemics. And all of us already understand what really it's really not a very virulent virus has done, the SARS-CoV-2 virus, COVID-19 pandemic, what it has done to the world within less than 12 months. And this is not even a highly virulent virus like Ebola or some of the other hemorrhagic viral diseases. And just within a few months, our entire world, national economies have collapsed. People are going to war. And all of this is going to compound and get more and more difficult for us as time progresses and the number of people increases on the planet. And so we have to start to understand how we can address some of the issues. And very importantly, when it's going to be too late to address these issues, because there is a point of no return where you get to the point where you have a complete depletion of natural resources to a point where it's no longer renewable and no longer sustainable. And so this idea of doing research and trying to understand the nexus and the integration of food and energy and water on our planet is critically important. Now, as an astrobiologist myself, I often get a lot of questions about, why do you care about what goes on outside of the Earth? We have so many problems here on our own planet. Why should we be spending millions and billions and trillions of dollars to explore the moon, to explore Mars, to try to figure out whether there's aliens on some planet, whether they be microbial aliens or intelligent life, when we have so many problems that we need to work on here on our own planet? And, you know, that is a very good question. And it's a question that anyone as an astrobiologist or someone who's in space exploration needs to be able to answer. because doing space exploration is extremely expensive, and it not only takes away resources from research on food, energy, and water, but it takes some of the most brilliant minds, some of our best scientists off of the focus from the problems we have on our own Earth, because they're busy spending time putting their focus on exobiology and astrobiology and space exploration. So these are really good questions, and we have to be able to answer them. And honestly, I don't know if we have a very good answer, but there are some answers, and I like to provide some explanations why space exploration and astrobiology is important. And one of the most recent ones is a renewed look at one of the largest asteroids in the asteroid belt, which is called Psyche. It's 16 Psyche is the actual name for it. And this is part of the asteroid belt between Mars and Jupiter. And it's about 140 miles in diameter. So, I mean, it's basically a small size of a small moon. And it was initially discovered in the late mid 1800s, but it's composed almost entirely of iron and nickel. It is estimated that this chunk of metal by itself is 70,000 times more valuable than the world economy. So it's worth 10,000 quadrillion dollars. So imagine how many zeros is 10,000 quadrillion dollars, right? And that represents about 70,000 times the world economy. And so space offers some opportunity to extract valuable mineral resources. And if we had that iron and that nickel available to us on Earth, then we would expend a lot less time, money, energy, water, because all of you know that mining metals requires a lot of water usage, fresh water usage, on mining iron and nickel in our own country and destroying our forests. So there's a lot of nickel mining in Honduras, in the jungles of Honduras, for example. And we would have this extraterrestrial resource available to us that would help us to preserve food, energy, and water while bringing wealth to our planet from abroad. And so this is just one example. Another example that I like to talk about is the fact that we are going to have problems with fresh water. Fresh water is already a problem with, ocean acidification, with, pollution, and again, climate change and farm runoff, which causes eutrophication and dead spots within some of our most productive ocean environments in terms of fisheries. We're not only having problems with our oceans, but the amount of fresh water is going to be limited as well. And if you look into space, of course, these are millions of miles away, but we have super large resources of fresh water that are available, not accessible, but available to us, not accessible yet, in our solar system. And I just use two examples. We take a look at two of the moons within our solar system, Europa, the moon of Jupiter, and Enceladus, the moon of Saturn. And just within these two bodies alone, it is estimated that we have enough fresh water for thousands and thousands and thousands of years. And so if you take a look at Europa, not only do you have a water ice crust, but it also has, sort of a core, a metallic core, and it also has a lot of silicate rock. And below that water ice crust, it is theorized that there's either a liquid ocean underneath, or sort of a movable ice, so soft ice, if you will, just to keep it as a simple explanation. If you go out a little bit further, about double the distance, then you start looking at Enceladus, one of the moons of Saturn, and there's very, very good evidence that there's water vapor actually emerging from the icy crust of this moon. And underneath that, of course, is theorized to be a huge ocean. We don't know the salinity of the ocean, but we imagine that a lot of the ice, both on Europa and Enceladus, is clean ice, so can be used for freshwater extraction. And so in our solar system, in space, we have resources that actually address two of the key issues that we're worried about, right? And that is precious minerals, energy, and water. And so in a way, space exploration, astrobiology is important to helping to solve some of the potential problems that we're going to see over the next couple of centuries here on our planet. And so I want everybody just to think for a moment about how astrobiology and this research into the innovations of food, energy, and water systems in terms of what's going on our planet are related. Now, one of the things that I do, and this is just some big thoughts, I want you to think about these big thoughts, is I'm interested in understanding how microbial organisms and microbial ecosystems contribute to food, energy, and water, and how we can build biotechnologies based on things like extremophile proteins to help solve some of the world's problems, right? And so one of the key things that I'm interested in is sort of the cellular physiology, especially in terms of photosynthetic capability of photosynthetic microorganisms. So we have both cyanobacteria and we have single-celled eukaryotes, microalgae in particular, that can do something that's just phenomenal that I wish I could do. They can photosynthesize. So they can take direct energy from the sunlight, so solar energy, and they can convert that to organic molecules that are essentially nutrients for everything else that's living on our planet. On the far right picture here, this is just a lawn of green microorganisms that have been infected by a virus. So these little plaque-like structures here are a cyanophage that is basically infecting this lawn of photosynthetic microbes, this cyanobacterium here. And one of the things that is becoming more appreciated now in my world, in the microbiology and virology world, is the critical role that viral mediated processes have in driving evolution, both of the host and the virus, and causing and helping to facilitate major transitions in the evolution of life. So when we talk about the origins and evolution of life, it's ridiculous to consider anything, any system, any theory without considering the impact of viruses, okay? Now people will say, well, that's just some little microorganism in a bacteriophage, and that's a legitimate excuse. But we can also think about a little bit more of a funnier example. And, you know, I'm talking this in kind of humorous way, but if you just think about let's say coronavirus, for example. So we're all in the middle of being locked down and isolated, can't go to school in some countries, in some states, because of the COVID pandemic. Now, lucky for us, the SARS-CoV-2 virus, that particular coronavirus, there are lots of coronavirus. So when I say coronavirus, I'm referring to SARS-CoV-2, is not as virulent as a virus could be. Now, that is not meant to diminish the deaths that have been caused by coronavirus. Of course, any death is sad and something to be concerned about. But if you look at the number of people who are infected with SARS-CoV-2 and the number of people who actually die from the viral infection, the percentage-wise, it's very, very low compared to something like in the past smallpox or Ebola virus outbreaks and these kind of highly virulent viral pathogens. And if you were to imagine in your head that SARS-CoV-2 is one of these more virulent pathogens, and we look, especially in the United States, we kind of poke fun at ourselves in some cases because as Americans, we're so like worried about having to stay in and not meet with our friends and be isolated. We can't handle it, and we get really upset from being isolated and not being able to go to work and school. But at the end of the day, if it's a matter of life and death, like it would be with an Ebola virus outbreak or something, those concerns and those issues that we have really pale in comparison to going out into the environment and dying, right? And so if coronavirus indeed was highly pathogenic, then you would take a look at your populations of humans, and those people who are very comfortable sitting in the basement of the home of their mom and dad and just playing video games all day, and they don't have a problem with sitting at home in a dark basement and playing video games, those would be the future of the human race. Because all the other people who can't stand to be enclosed and be in home would go out, they would get infected, and they would die. So you can imagine over the course of multiple generations, the human population would change from Homo sapien sapien to this Homo sapien laptopticus, right? The guys that like to sit and work on the computer in the basement in the home of their parents. And so this idea that the evolutionary trajectory of species is highly dependent upon the interactions between disease agents. So viruses and even bacterial and fungal infections cannot be dismissed. And so when we think about all of these challenges with food and water and resource limitations, we also have to think about how not having those resources available will automatically increase the amount of emergent infectious diseases. And of course, viral diseases and bacterial and fungal diseases are a lot of the germ-based diseases which would dominate the system. Okay, and so thinking about viral infection and virus-host interactions is extremely important on Earth in terms of evolutionary trajectories of species. And we're very anthropocentric. We like to think about humans first. So it's super important for humans and human survival. I would argue that plants are cooler than humans because they can photosynthesize, even though I'm not a plant biologist. But since we are anthropocentric and we like to think about humans first, you know, we need to think about how virus-host interactions and other types of pathogenic diseases are going to emerge when we have resource limitation. We can see how virus-host interactions and virus-host dynamics would work in what we call reduced model systems. So looking at things like bacteriophage bacterial systems and even archaeal virus systems can give us some insights and some fundamental rules about how higher order species, multicellular organisms, including mammals and humans, might react to different types of viral infections. And so even though I do astrobiology and I do some biomedically related work in my lab, sort of the big overarching question that I have, you know, like when I retire, what I want to say that I've contributed to is the hopeful development of a set of fundamental laws that explain how virus-mediated processes influence both host and virus evolutionary trajectories over long periods of time. Now, because the virus sphere is quite diverse itself, meaning we have many different types of viruses and potentially different points of origins of viruses. So, you know, the RNA viruses, the segmented RNA viruses, the single-stranded DNA viruses, the double-stranded DNA viruses, we don't really know the different origin points of those. We have some idea of lineages, but like in cellular life, where there's this idea of a you know, last universal common ancestor, within the virus sphere and within virology, there are quite a lot of people who believe that viruses emerged multiple times over evolutionary history. And if you look along these different lineages, then you're going to find different points of origin. And so in my lab, I have to narrow it down because viruses are just so diverse. So my lab focuses on double-stranded DNA viruses. But what I do is I look across domains of life. So I have cyanophage and cyanobacterial systems that I look in, and we're sort of working on trying to understand Phycodnaviridae. There are other people who do a lot more and better work than we do with algal viruses at this point, but we're interested in those double-stranded DNA viruses. We look at double-stranded DNA viruses in archaea, which are shown here, and I'm going to spend a little bit of time talking about today. And then we also work in double-stranded DNA viruses in mammals. So I look at a roseola herpes virus in mice, and I also look at a very, very common herpes virus called human herpes virus 6, or HHV6, which 99% of the human population is infected with by the age of 3. All of us, by the age of 3, we get an initial viral infection with human herpes virus 6, and we get a little rash on our limbs and our trunk. And then this virus goes latent. And most people live with the virus for their entire life asymptomatically. There are no symptoms. However, there are cases in which that virus can reactivate during adulthood and cause neurological dysfunction like multiple sclerosis, and we study the potential for HHV6 to cause epileptogenesis or epilepsy. However, those complex systems, human systems and mammal systems, we don't have a set of fundamental laws like we have in physics. I did my undergraduate degree in physics, and I always tell my students, physics and chemistry to me is simple. Biology is complicated because in physics and chemistry, physics, for example, you have Newton's laws, you have the laws of thermodynamics, you have Schrodinger's equations, you have Maxwell's equations, you have these fundamental laws. And so for any problem that is presented, you can go back to those fundamental laws and you can derive a potentially reasonable solution. But in biology, which is a combination of chemistry and physics coming together, chemistry and that biological systems are based on a cell, which is matter, biomolecular structure and function and architecture, and energy in terms of the chemical energy that's required to drive those cellular systems. Biology is complicated because there's so many unknowns, and it's so difficult to predict what's going to happen in biological systems. And so what I would like to do is understand, just for the double-stranded DNA viruses, are there fundamental laws that we can develop that will help us to predict the evolution of virulence, or on the other side, help us to estimate when a virus will attenuate during a pandemic or some kind of infection. Since it's difficult to do that in macroorganisms, I study microorganisms. So what you're seeing on the screen right now is what's called the Ceballos spindle-shaped virus. And so here are 4 virus particles that are bound together at the hydrophobic tails. And my lab spends a lot of time studying how this spindle virus infects its host species, Ceballos, which is a hypothermophilic archaean that lives in volcanic hot springs around the world. And we also are designing biotechnology, which helps in food and energy. And the hosts for these viruses, so this is the host here, this is Ceballos, this is a cross section. I'll show you a full cell in just a minute. They also express chaperonin proteins, which are thermophilic heat shock proteins, which are shown here in gray. And in collaboration with some colleagues at NASA Ames Research Institute, who developed a fusion protein, and I'll talk a little bit about this in a minute, we can bind enzymes to this thermophilic platform, and these enzymes can be protected in extreme environments, like extreme acidic environments or high temperature environments, because that's the natural function of this heat shock protein complex in the thermal hot springs, in the volcanic hot springs. And so I want to spend a little bit of time. I have, maybe 10 minutes here, and then we'll take some question and answers, talking about these 2 lines of research. So this Ceballos spindle virus here infects this Ceballos, which is a hypothermophilic archaean. And this hypothermophilic archaean also expresses heat shock protein complexes. And we've used these heat shock protein complexes for biotechnology, both for enhancing lignocellulosic feedstock deconstruction, which would be a new wave for bioethanol, so we can avoid the food versus fuel dilemma. So we don't have to use corn or sugarcane, the things we need to eat. We can use the leftovers, the byproducts. And we are now using this system, testing the system in chicken feed, so that we can help what's called food conversion ratio, which is using less feed and less water to grow a chicken to harvest weight in a shorter amount of time, by protecting the enzymes that are put into the chicken feed. So you can see how astrobiology, the study of, the potential for extreme life that's found on this planet, and the possibility that these types of life forms might be found on other planets, can also lead to biotechnology that can help in our dilemma with food, energy, and water technology. And so here, I'm not going to go through this entire presentation because we don't have a lot of time, but these are hypothermophiles, and they're found in volcanic hot springs worldwide. Viruses have been found to exist with these microbes. They basically live in boiling battery acid. They live at temperatures anywhere from 65 as high as 90 degrees C, and pH is sometimes lower than 2. When I did some sampling in El Salvador in 2008, the pH meter registered less than 1, and I was able to extract Sophilobales from that volcanic hot spring. All of the volcanic hot springs except Italy, I believe so far, that have been studied, at least by my lab, also have viruses, these spindle viruses, which infect Sophilobales. And we want to understand, sort of how these virus-host interactions work from a microbial ecology standpoint and astrobiology standpoint, so that we can start to develop those fundamental laws that I talked about earlier. Now, because these volcanic hot springs are found all over the world, we have something called biogeographic barriers to dispersal. So you would think that if there was a Sophilobus and a virus here in the Azores off the coast of Portugal, that these are islands and there's a big ocean between. You probably won't find the same genus or species in, say, Iceland, which is another island that's separated by a large distance, or in New Zealand, or down here in, sort of the southern tip of South America in Chile or Argentina. But you know what? We do find very, very similar species, some the same species, definitely within the same family, definitely within the same genre. And so it's amazing to us that we can find hosts and viruses from these very distinct geographic regions. And what's even more, kind of the 8th grade high school idea is, okay, well, you know what? I want to take a virus from here and I want to try to infect a host from over here. Or I want to take a virus from here and I want to try to infect a host from over here. Just to see what the coevolutionary landscape looks like between virus and hosts that are endemic to this Iceland volcanic hot springs versus El Salvador and Costa Rica versus Italy. And we did that work. And we went out and we took samples from all of these different volcanic hot springs, and we started doing these cross-infection assays. Here's an uninfected cell, here's an infected cell, again, an infected cell, and we started looking at which viruses can infect which hosts and what is the relative virulence. You'll see here that there's a high virulence with this virus on the same host lawn, and there's a very lower virulence here almost no virulence of SSV10, which was found in California on this host that came from Italy. And so we found a couple of different patterns. Some hosts were completely susceptible to all the viruses, some hosts were resistant to all the viruses, and some hosts were only susceptible to 1 or 2 of the viruses. And so we published this study in 2012, and we started looking at a couple of the viruses and their relative virulence to try to understand these fundamental properties. And we came up with some interesting information about non-lytic viral replication versus lytic viral replication. And I don't have time to go into the whole story, but this is published in Frontiers of Microbiology, and you can take a look at that. So we have these different viruses that come from different volcanic hot springs. Why do they have different levels of virulence? And so that's a question that we want to know. We recently published another paper in Frontiers of Microbiology looking at one specific virus from Russia that tend to kill all of the hosts, no matter which part of the world they came from, except a host from Russia, in which case it made it sick but it didn't completely kill it like you see over here. So this is part of what we've done. And then in collaboration with my folks at NASA Ames Research Center, we've taken, let's move away from the viruses. We've taken proteins out of these hosts. So we've taken heat shock proteins, and we've developed biotechnology to basically tackle some problems that we have in bioethanol and now in chicken feed by combining a domain, a cohesin domain from another bacterium called Clostridium thermocellum. So this little yellow area here, we stole that and we attached it to our platform to make this sort of Death Star Medusa Pac-Man scaffold. And what we can do, so here's the protein, here's the original protein from the archaea. Here's the protein from Clostridium thermocellum. We can bind enzymes to that, and those enzymes can be protected under harsh conditions of low pH or high temperature. And we're trying to apply that to solve some problems with food and energy. So with that, I think I'm running short of time. I would like to stop right now, and I would like to take questions from the audience. So thank you.
Thanks so much, Michael. Wait, my video. Wait.
I don't see you.
No.
There you are.
Michael, thanks so much. It's an amazing presentation. I speak with you in Spanish because I think it's so easy for our host. Michael, muchísimas gracias nuevamente. Una presentación maravillosa, una propuesta excelente de todo lo que nos comentas. Tenemos muchísimas preguntas. Voy a intentar resumirlas e integrarlas lo máximo posible para que nos los puedas ir comentando. Una de las principales cosas que nos preguntan y que además es muy relevante de tu propuesta, de tu presentación, es todo lo relacionado con la energía, el agua, la comida y las problemáticas con las que nos estamos enfrentando ahora en nuestro planeta. En los últimos años se han desarrollado algo que nos comentan, que son los Objetivos de Desarrollo Sostenible de la Agenda 2030. Y de los principales retos que tienen esos objetivos es todas estas problemáticas que están relacionadas sobre todo con la contaminación del agua, con los efectos que nos encontramos en nuestro planeta, el calentamiento global, la comida. Y como sabemos que estás haciendo muchos proyectos, que sé que todos son maravillosos, nos gustaría que nos comentaras de pronto uno o dos que te parecen que son como muy especiales o relevantes para que los compartas con nosotros, porque nos quedamos con las ganas de que nos cuentes todos los proyectos. Sé que tenemos poquito tiempo y nos hace falta como que nos cuentes todo esto, pero nos gustaría que nos compartieras un poquito de algunos de estos proyectos que están realizando en Arkansas, que nos cuentes un poco, porfa.
Pues sí. So one of the things that I like to consider is the fact that if you think about extremophiles, which is one of the big areas in astrobiology, right? Extremophiles are interesting in themselves because they have already survived, even though they're only single-cell organisms, they have already survived through periods of resource limitation. They can survive in harsh environments, so they can survive in acid and high temperatures. And so there's a lot that we can learn from these microorganisms. But in addition to that, we can basically steal things from those organisms. And so one of the projects that I have going on right now, which I think is incredible, is the one that I talked about, which is trying to understand how we can use thermotolerant and acidotolerant chaperoning complexes that have the natural role to protect other enzymes and proteins in these hypothermophiles during occasions of temperature fluctuation and pH fluctuation. So, en la naturaleza se funciona para proteger todas las proteínas en la célula cuando hay problemas con temperatura y pH. And if we can take that scaffold and utilizarlo para la industria, para resolver problemas de energía, resolver problemas de comida, de producir comida, sería excelente. And right now, we're working on engineering these scaffolds to help the poultry industry so we can produce good quality food using less water and less feed, and also maybe applying this to bioethanol production so that we don't have to depend upon corn-based or sugarcane-based ethanol, which produces a food versus fuel. So alimentación contra biocombustibles or combustibles, they can overcome that problem. Now, for you guys, one of the things that's really interesting is that we have a really strong collaboration with the Astrobiology Institute of Colombia. And IAC has programs where students and faculty alike can come to the United States, and we have Estación del Campo, we have a field station where you can come and you can engage in research in microbiology or even fish genetics, or it depends on where you want to work. If you want to work at the microbial and molecular and cellular level, or if you want to work at the organismal level, we have some programs. The IAC has programs that you can participate in to get your foot in the door and start learning how to do research in some of these fields. And I hope that all of you will take time to look at the AIC website and see if any of those programs might fit you. So those are some of the things that I think are interesting. Espero que contesté tu pregunta, su pregunta.
Genial, maravillosamente. Muchísimas gracias. Claro que no la contestaste, pero pues con lujo de detalles te lo agradecemos un montón. Otra de las preguntas que nos hacen es de estos modelos que estás realizando con arqueas, ¿cómo crees que con las arqueas, con estos procesos evolutivos que se están desarrollando a través de los virus, cómo consideras que se pueden encontrar, por ejemplo, en algunas zonas como las Islas Azores o Nueva Zelanda, las que nos comentabas, de estos proyectos, que varios de estos modelos que nos has ido comentando a lo largo de tu presentación, que son muy relevantes y realmente a través de la biotecnología pues están marcando la diferencia, porque efectivamente nos están mostrando cómo, por ejemplo, organismos como los hipertermófilos como tal, o los que están muy asociados efectivamente con volcanes, nos pueden dar una luz para ver este proceso evolutivo.
Yeah, and so there's, well, as you know, I mean, there are different types of extremophiles, right? So there are folks who spend a lot of time studying halophiles, so microorganisms that can withstand a lot of salinity. And then there are psychrophiles, so organisms and microorganisms that live basically in ice or in small salt veins within ice structures. And then there are the thermophiles, like the ones that I study, that live basically in boiling battery acid. You know, I mean, it's boiling and it's acidic, it's hot, right? And so if you think about what really drives life, so what's really important in our cellular systems is enzymatic activity, because life is about a constant set of reactions. That's what keeps cellular function ongoing. And those reactions are highly regulated, and sort of the policemen, if you will, that allow those reactions to go. You can think if you're at a traffic light and driving and you have the police officer saying, okay, this cars move this way and those cars move that way, right? In the cellular world, those are the enzymes, right? And so protecting enzymes, understanding how enzymes can function at very low temperatures and how enzymes can still survive at very high temperatures and low pH, in my view, is key to understanding the possibility of life on other planets and other moons where those sort of conditions may exist. And again, we know that the basis of life, the basic unit, is the cell, but those cells are not going to evolve at an appreciable rate without the activity of some type of virus type entity, whether it's a viroid, a bona fide virus, or some other mechanism that resembles a virus-like activity, so that you're not just relying on single nucleotide point mutations, but you're seeing huge chunks of genetic material moving through the environment so that species can evolve at an appreciable rate. So I hope that answers your question. Espero que he contestado.
Of course, muchísimas gracias. Y bueno, Michael, lo que te comentaba, tenemos un montón de preguntas, pero bueno, que los estoy tratando de integrar todas para poder como cumplir y resolver preguntas. También, primero, si alguien quisiera hacerte preguntas o tiene dudas o quiere conocer más sobre tus proyectos, para que nos lo digas ahora al final, dónde pueden contactar, o la página de los proyectos de la universidad, o bueno, de tus artículos, que además son absolutamente maravillosos, que he tenido el placer de leerlos y son maravillosos, para que nos comentes ahorita al final. Una última preguntita que está relacionada con las propuestas y los proyectos que se están haciendo en la Estación Espacial Internacional, que también se están llevando de alguna manera dentro de estas condiciones controladas, organismos extremófilos, o se están haciendo proyectos y propuestas, incluyendo la biotecnología, para resolver o bueno, investigar también en diferentes condiciones, ya no terrestres como tal, que son como las que tenemos un poquito más al alcance, sino en otras condiciones. ¿Hay alguna que quisieras resaltar o que nos quieras comentar un poco más sobre estas propuestas maravillosas de extremófilos, de esas propuestas en la estación?
Yeah. So I'll answer your first question. La primera pregunta primero. Okay.
Dale.
So si una persona quiere saber más de nuestros proyectos, pues hay dos maneras. La primera manera es para preguntar al Instituto de Astrobiología de Colombia, porque tenemos un acuerdo con ellos y ellos tienen programas. Ustedes pueden venir. Una persona puede venir aquí con nosotros. Tenemos una estación del campo en Minnesota, que es una hora sur de la frontera con Canadá. Entonces, en los inviernos, es todo hielo y nieve. Todo hielo y nieve. Y podemos ir a sacar muestras de sedimento, de lodo, de soil, y podemos ver si hay organismos, microorganismos que todavía están funcionando o si están como dormant, right? And we also también tenemos programas en el verano. Right now, ahora por las restricciones por el COVID, no podemos viajar mucho, pero cuando todo eso pase y tenemos la oportunidad para viajar otra vez, sí, tenemos muchas oportunidades. He viajado mucho por todas partes del mundo, de Asia, Malasia, Vietnam, ¿dónde más? Tailandia, Europa, España, Portugal, Noruega, y buscamos ambientes donde podemos encontrar microbios extremófiles, y pues hay oportunidad. Entonces, la otra pregunta sobre un sistema espacial.
Sobre la Estación Espacial Internacional. Sobre proyectos de tecnología
Sí, estación espacial.
Yeah.
Oh, ok, yeah. Well, pues trabajé en los 90s. En las 90s, in el Boeing company. Boeing was the primary contractor for NASA Space Station. And what I did there, I started off working in what's called flight modules design. So there are end cones on the modules. Los módulos tienen end cones, las capas, they're caps, right? Y I worked there como ingeniero. I mean, I was doing engineering work, not biology. It was interesting. And I was packing the systems in these end cones. But one of the things that really gave me a lot of interest was the environmental control and life support systems. So we had to package these. And I didn't know anything about biology, but one of the things that really interested me was this idea that you have to protect the astronauts against bacterial and virus infections. Because once those things are, I mean, this is a closed system, right? So you're not going to be able to get rid of it. Like here, you know, abre las ventanas, open the windows and let the sun in and spray everything down. No se puede, no? All right. And so we had to have filtration systems and air conditioning systems and trying to consider what happens if you have a mini pandemic inside the space station. How are you going to deal with that? So that really piqued my interest in environmental control and life support systems. And it helped me to get into biology and make me wonder, how do viruses, how do bacteria not only affect people in active disease states, but even if they're very low virulence, where there's no apparent disease, like neurotropic viruses. I study a herpes virus that infects the brain, but it doesn't cause any notable disease, but could it cause changes in behavior, right? Even if it's low production. And so when you're in space, whether it's in a space station or whether it's on a lunar colony or a Martian colony, the interaction between microorganisms that are potentially pathogenic and the folks that are playing on the moon or in Mars or working, you know, in some, you know, celestial body, that health has to be taken into consideration. And so there's a lot to be said for microbiology, both here on Earth and in space. Otras preguntas.
Muchísimas gracias. Bueno, primero, agradecerte sobre todo lo que nos comentabas, bueno, todas las preguntas, todas las respuestas, agradecerte por lo que comentas con los proyectos que tenemos de la mano del Instituto de Astrobiología de Colombia, que estoy segura que es de gran interés para absolutamente todos y te vamos a estar contactando para que podamos hacer este tipo de convenios, este tipo de proyectos que estaremos encantados de trabajarlo contigo. Y ya solamente la última preguntita, porque de verdad que, como les decía, es que son temas tan apasionantes que nos quedaríamos aquí todo el día. Nos hablabas de las cianobacterias y ya, como te digo, para terminar, se han hecho algunos experimentos, algunas propuestas en las que se utilizan cianobacterias o se utilizan también algún tipo de bacterias específicas que ayudan a generar algo de energía o bueno, que se están implementando a través de la biotecnología. ¿Nos podrías contar algo de eso? O?
Pues sí, hay dos como caminos que hemos probado. La primera es investigar los fagos, los viruses que mueven los genes involucrados en el fotosíntesis entre especies distintas de cianobacteria y también entre los cianobacteria y los eucariotes, los microalgas, right? ¿Cómo pasó esta transición entre los cianobacteria y los eucariotes, los microalgas, ¿no? Entonces, saber esto es importante, pero por lado de la energía, fotosíntesis, y no soy experto en fotosíntesis, pero yo doy todos los, I give a lot of credit to everyone who studies photosynthesis because photosynthesis is amazing. You take basically carbon dioxide, un molécula muy pequeño, y luz, y puedes crear azúcar. You can make sugars, right? Organic molecules, and that's absolutely incredible. So I think there's a lot of technology out there to use photosynthetic microorganisms to produce biocombustibles, so bioethanol and biodiesel. There's a lot of new work that's interesting looking at what they call extracellular electron transport. So we have electron transport chain in mitochondria, but some microorganisms can use, especially those that live in clays and in other types of sediments, can have an electron transport on its extracellular, in its plasma membrane space. And they can do redox reactions and generate energy based on what's in their environment using sort of extracellular electron transport. And that's pretty exciting. So there's a lot of different exciting work out there. But one of the things I wanted to mention too, with regard to these programs, so the Institute for Astrobiology offers programs to students and faculty. But the other thing that I want to mention is that these programs are just one step in the door. If you're really good and you're really excited about the work, we have un convenio with astrobiology. You can apply to the University of Arkansas for your master's degree or your PhD program. And what a lot of students don't understand is that you get paid. When you are in a master's degree or a PhD program, it's not like bachillerato, like the first carrera, la primera carrera. When you're doing bachelor's degree, you have to pay la matriculación, you have to pay, go to class, and then you get your degree. When you're a PhD student, graduate student, you get paid. It's not a lot of money. You're not going to be rich, but se puede sobrevivir. You can have enough for an apartment with a roommate. You can have food. And so there's a lot of opportunity if you're really interested in studying microbiology or evolution, microbial evolution. There's a lot of opportunity. The first step in the door is to make that investment into the programs that are offered by the Astrobiology Institute. Cost a little bit of money, but it's an investment. You will get that return on the investment later when you enter your master's degree and your PhD degree. So these are great opportunities, and I hope that folks will take advantage of that.
Michael, muchísimas gracias. Again, de verdad, absolutamente maravilloso. Te doy las gracias por parte de todos los miembros y todos los asistentes al Congreso, porque realmente no solamente con tu maravillosa ponencia, sino además con todos los proyectos y propuestas que nos comentas, tanto para maestrías, para doctorados. Te aseguro que estamos muy interesados. Agradecértelo nuevamente por esta maravillosa charla. Y bueno, nada, me toca despedirte. Pues un abrazo, espero que nos veamos pronto. Y bueno, voy con el máster.
Nos vemos, gracias.
Nos vemos, bye.